Let's revisit the central dogma of molecular biology. A DNA sequence or gene is transcribed into the mRNA, which is then translated into protein. In a simple term, a gene contains start and stop codons within the DNA. For instance, the start codon ATG signal the beginning of the translation and codes for methionine in the resulting peptide. The stop codons, such as TAA, mark the end of the translation. For reference, we often use a cheat sheet to look up this codon and their corresponding amino acid. This foundational concept provides us with a biological framework for identifying the location of a specific gene. A basic algorithm can be used to find gene region, search for the start codon ATG, then look for a stop codon such as TAA, TGA, and TAG, ensuring that both are in the same reading frame. This means that the sequence between start and stop codons should be divisible into a group of 3 nucleotides codons. Using this approach on a sample genome, we might detect 2 potential gene regions. While this is a straightforward method for identifying a gene candidate, it's important to add biological context. For example, genes typically encode peptides longer than this unreal sample genome base, so very short sequences are unlikely to represent the real genes. Beyond identifying gene coordinates, the next step is to determine their function. Proteins were studied and cataloged long before the advent of DNA sequencing. An intensive protein sequencing database exists. By comparing newly identified DNA sequence to non-protein sequence, we can infer the gene function, especially when there is a strong match to a characterized gene, even across different species. Today, thanks to advances in DNA sequencing technology, we can more accurately map a gene location and explore alternative transcripts, revealing how exons and introns are combined in different ways for specific genes using RNA sequencing that I will explain in the next topic. This ongoing research continuously updates on our understanding of gene structure and function.